So if you remember last day, we had introduced prokaryotic cells. We talked a little bit about the differences between prokaryotes and eukaryotes. And I also made a few notes on archaea. So that's almost everything we're going to say about archaea for the semester. As I mentioned, we know a lot less about them, and we know a lot more about bacteria. So this is where the focus is going to be for the rest of the topic. And when we talk about prokaryotes for the rest of the semester, for the most part, we are talking about bacteria. So let's get into this. What do bacteria do? They do all sorts of things. We find them everywhere where we look. We find bacteria. Many of them live inside our body and on our skin. E. coli is a very good example of that. E. coli lives with us. It lives in your intestines, and actually it's very, very happy there. You have a warm body. You have lots of moisture. We stuff our faces with food. And E. coli is very happy to live with us. And as a consequence, what does E. coli do for us? Well, it actually makes niacin. That's one of the B vitamins. And I think some strains actually make the vitamin K. In some cases, it actually helps us digest a little bit of lactose. For the most part, E. coli is good. I know a lot of people have been trained to think of E. coli as bad, and we're going to talk a little bit about E. coli and why E. coli is good. Kind of at the end of this topic. So we might not quite get to it today. It might have to wait till Friday, where we're going to talk a little bit more specifically about E. coli. Some bacteria are very nasty. You may or may not know about this one. This is Yersinia pestis. Yersinia pestis causes the plague. So this was a very important organism. Historically, you can see on the right there, I have a diagram of a plague doctor. And you can look all about the Black Death that was in the 1300s and all of the plague epidemics that happened in the last 400 or 500 years, just absolutely decimating populations in Europe and Asia and various parts of the world. Plague, very bad. About 85% of the infectious diseases that are known to humans are actually caused by bacteria. So they're very, very important medically. Bacteria are also very important because many of them are photosynthetic. This here is a diagram of cyanobacteria. So you may or may not know this word here, cyan. Cyan kind of means that bluish, this color here. Kind of this bluish color. It's sort of kind of like turquoise or teal. If you ever drive along the highway and you see a swamp with that color in it, that's what you're looking at, mostly cyanobacteria. These things are producing loads and loads of oxygen, maybe half of the oxygen on the entire planet. So they're very, very important for us. Plus, they're a big part of the food chain, of course. So very, very important. Many bacterial decomposers and involved in nutrient cycling. This is a diagram of rhizobium. These are involved in nitrogen cycling, and you can see they're actually found on these root nodules in legumes. And many, many, many more things. You can see I've got some notes here about bacteria involved in food production. If you like yogurt, that is a bacterial culture. They're involved in biotechnology, and they're involved in all sorts of aspects of human nutrition, all sorts of good things. So bacteria are good. I hope you guys like bacteria, because I certainly do. So for today's lecture, we're going to go over a bunch of these structures. You can notice this is a prokaryotic cell. There's no nucleus. But there are quite a number of different types of structures that are important for the bacteria. And many of these structures are actually really important for this organism to cause disease. So we're going to kind of focus on a little bit on disease kind of at the end of this topic. For now, we're just going to go over what all these things are. So first of all, there's the cytoplasm. You probably know a little bit about the cytoplasm. We've talked about it already. Remember, we said cyte means cell. And plasm means goo or juice or something like that. And so this is the goo inside the cell. Usually we think of the cytoplasm as kind of this big empty void and mostly water and all that. And I wanted to show you this diagram here. This is drawn by a guy named David Goodsell. And he's a biochemist, and he's also an artist. And so he draws these things and he tries to make it accurate to the actual biology. And I really like this diagram because he's showing you that the cytoplasm is indeed full. It's not just water, it's full of all sorts of things. So some of these things are labeled in my diagram here, and he's trying to make them, like I said, accurate to what they should actually look like. So these big purple things, these are ribosomes. Ribosomes are, of course, made of RNA and proteins. And you can see this messenger RNA is being made by the ribosome. So let's talk about ribosomes. I'll come back to that picture in a minute. So ribosomes are where translation takes place. Translation is protein synthesis. I'm going to make a note on the ribosome in a minute. I'm going to make another note document talking about these bacterial structures. So if you want to get a piece of paper and pen, we'll come to that in a minute. So I want to show you that prokaryotic ribosomes and eukaryotic ribosomes are different. So I mentioned this last day in that table that we made that the prokaryotic ribosome was 70S and the eukaryotic ribosome is 80S. So what does that S stand for? S stands for Svedberg unit. So that was actually a Swedish guy, and the Swedish guy was doing some centrifugation, and he found that that eukaryotic ribosomes, they sedimented much more quickly than prokaryotic ribosomes. Eventually, he made a scale around how quickly things sediment, and it was named after him. S, you can think of as Svedberg, or you can think of it as sedimentation as well. And he characterized these two different types of ribosomes. So eukaryotic ribosomes are bigger. If you take a look at ribosomes, they have two subunits. There's a large subunit and a small subunit. And each of these subunits are made of various RNAs and proteins. And the composition between eukaryotes and prokaryotes is different. This is actually really important if you've ever taken the drug tetracycline. Tetracycline, it turns out, will bind a bacterial ribosome, but not our ribosomes. So it's a good drug for that reason, in that it will kill the bacteria and not harm the host. Okay, so I am going to start a Word document here, and there it is. And you can see we're going to make some notes about bacterial structures. So you can see the first one I have on there is the ribosomes, and we just mentioned what ribosomes are made of. So I'm going to have a ribosomal RNA, and they're made out of proteins. So what is the function of ribosomes? Ribosomes are the site of protein synthesis. So that has a name, and that is called translation. Okay, so I'll be coming back to this document again and again. So if you miss something, don't worry, we will come back to it. Or you can always review the lecture videos if you miss something as well. All right, back to our PowerPoint and back to our diagram made by David Goodsell. So he's showing what he's seeing in the cytoplasm. These little blue things, the smaller blue things, are actually proteins. He's got some of them labeled here. You can see enzymes and proteins, all sorts of different ones in there. Each of them represents a different protein or a different enzyme. And the yellow spaghetti-like stuff is the DNA. And if you look closely, what he has there right in the middle is he's showing DNA polymerase. So DNA polymerase takes one strand of parent DNA and makes two daughter strands. So you can actually see that right there in the diagram. So kind of cool. He's thought of everything. Well, not quite everything, but it's a very nice diagram. So let's talk about the DNA. We don't have a nucleus. There's no membrane-bound nucleus here. But what we do have is a chromosome. In fact, in the diagram on the top, you can see there's two chromosomes because this particular organism is in the middle of dividing. So it's already replicated its chromosome. So you can see in my note there, it says the chromosome is a single loop of DNA. So the word loop is a little misleading because it's not like it's a big hula hoop. It's a little bit more like a twisted up rubber band. So rather than being a big loop like that, it's a bit more like this. And it's folded up, but it does join. So it is circular. There's one chromosome in bacteria usually. There are a few exceptions, but for the most part, this is the case. We've got one single circular type of DNA. So there's another term I wanted to show you here. You may notice it says nucleoid region. So what does that mean? There's no nucleus, so what's a nucleoid? Well, the DNA does in fact kind of clump together, and you can see this actually quite visibly on an electron microscope. And so the electron microscope people, they decided to give it a name. You can see it very visibly, and I don't know why they didn't just use the word chromosome. We already had a word for that, but they called it the nucleoid. So make sure you do know that particular word. The second thing down here at the bottom I want to show you are something called plasmids. So you can think of a plasmid as kind of like a mini or a super mini chromosome. So in E. coli, the main chromosome has about 4,400 genes, so 4,400 genes. If it has a plasmid, the plasmid might have 2, maybe 5 genes, kind of typical. There are some with more, but 2 to 5 genes is pretty typical. So it's kind of like a little extra chromosome that has a little bit of extra genetic material that it may help you or help E. coli to get along in the environment. So I know I have a little table here that shows you some of the genes found on plasmids. I'll share that with you in a second. I want to show you this diagram here. This is a really cool experiment. And this is an electron micrograph image. And what they did was they used some sort of detergent to gently, and enzymes to gently digest the cell membrane, proteins, and the cell wall. And slowly the genome, so the DNA was what was intact, and it kind of unspooled, and you can see it's all over the place. So not folded up and tight, but kind of spooled all over the place. And you can see this particular organism has a couple of plasmids as well. So that's kind of cool. So what are these plasmids doing? I mentioned that they're helping them survive sometimes, and here's a list of some traits coded on plasmids. And number one, this is the one we are most concerned about. It's worth it to put a little star beside that, because some organisms are getting drug resistance on these plasmids. So it turns out that some of these organisms can actually share their plasmids as well. So you may have a resistance to one drug, and it's shared between different species. And so this antibiotic resistance is kind of scary. When you have new genes that are discovered, scientists are watching them all the time and trying to understand what the implications are going to be for the medical community. There's a few other things here. You can see synthesis of pilus. I haven't talked about what a pilus is yet, so that's maybe not worth it to mention at the moment. Toxin production. So again, sometimes there's extra genes that are allowing these things to be pathogenic, which is a bad thing. So this is something we're concerned about when we see these plasmids. Okay, I know this is back to this diagram, but I am going to go back to the Word document and make a couple of notes here. So structure number 2 is the nucleoid. There we go. And what is the nucleoid made of? It is made of DNA, and I will put and associated proteins. There are some proteins that are found in the nucleoid region, and they're kind of organizing it, and sometimes you find proteins involved in DNA synthesis and things like that as well, if you're to extract the nucleoid. So what's the function of the nucleoid? This is, of course, the genome. So instructions for the cell, we'll say. Not the cello, the cell. There we go. All right, third one is we have plasmids. Okay, so what are plasmids made of? They are made of DNA. And what's the function? Extra genes that may help in the organism's survival. Maybe I'll put in bracket, scientists are concerned about genes for antibiotic resistance. Okay, so back to the PowerPoint. Like I said, if you missed this, don't worry. I will come back to it for the next item. So we're back to our our diagram here, or a little piece of art by David Goodsell, and you can see I have the last parts on this labeled. So some of the cell surfaces. So we talked about cell walls and cell membranes already, and you can see from this diagram here that E. coli is in fact gram-negative. Remember, gram-negative has, organisms have two membranes. So it has a plasma membrane or inner membrane, and it's got an outer membrane. And in between that membrane is sandwiched the peptidoglycan. So you can see I have that labeled there on the diagram. The outer membrane has those extra lipids, and you can see they're drawn as kind of those little fuzzy hairy-like things. And you can also see on there that David has drawn a flagella. So flagella is an appendage used for swimming. So we'll talk about flagella in a few minutes here. Okay, so kind of back to way back, I just wanted to point out here what is going on in this cell. This cell is dividing, and there's some terminology for you there, binary fission. So let's talk about that. Okay, so binary fission. What is binary fission? Binary means two, and fission means divide. Okay, so divide into two. It's not really a very great word. In fact, most scientists will actually just say bacterial cell division, but binary fission is stuck in all the textbooks, and you see it in the literature. It's basically a way to just say these things divide in two, and it's not as complex as something like mitosis. So let's take a look at these diagrams here. You can see we have E. coli there, and E. coli is just starting to divide its DNA. As you can see there in that little green part that's labeled, it says 2 copies of the origin. So notice this other note here. It says the chromosome attaches to the membrane. So think about mitosis for a moment, right? You probably know about mitosis, and you've got all those stages. You've got the prophase and the metaphase and the anaphase and the telophase, and you've got these spindle fibers, and the spindle fibers are grabbing the chromosomes, and they're doing it in a meticulous way so that half the chromosomes end up in each daughter cell. There's nothing like this with a bacterial cell. There's no spindle fibers. But they don't need them. There's only one chromosome. So we just have to separate the 2 copies. So how do we separate the 2 copies? The chromosomes actually attach to the membrane. And so watch this diagram here. You're going to see what's happened is it attaches to the membrane at the end of the cell right here. And as the cell stretches out, it's going to pull the 2 chromosomes apart from one another. So you can see that's what happened there. The 2 chromosomes are separated out. Once it gets to be kind of long enough, eventually the cytoplasm is going to pinch. So you can have something here called a cell septum. And the cytoplasm is going to pinch, and you'll get the cell membrane, the cell wall are going to separate from other, and you'll end up with 2 cells. So not as complex as mitosis, but it is something that does occur in bacterial cells. You need to know this terminology. Now, I think I have a couple of videos here. Here we go. I'm just going to take away the volume on that. So a couple of videos here. You can see we have some rod-shaped cells, E. coli, in the top video. And they're stretching and growing, and then if you watch carefully, you'll see them kind of pinch off and separate into 2 daughter cells. On the bottom, we have streptococcus, and streptococcus is growing and dividing kind of in a similar way, except for the cells are not rod-shaped, the cells are spherical or coccus-shaped. So that is binary fission. Okay, quick test question for you, some true and false questions. First one, true or false? Bacteria divide by binary fission and archaea divide by mitosis. Oops, sorry. The answer here is false. So both bacteria and archaea divide by binary fission. Archaea are prokaryotes, and they do what bacteria do. Question 2, the nucleoid region is surrounded by membrane. That is also false. Of course it is. We know all about prokaryotes now. We know they do not have a membrane-bound nucleus. Number 3, prokaryotic ribosomes contain 60S and 40S subunits. So you have to look that one up. but the answer is false, what the size of the subunits are. So there's just a quick comparison of binary fission with mitosis, and you can see all the stages of mitosis there. We can identify these stages. We have, it looks like prophase here, metaphase, anaphase, telophase, and then I guess cytokinesis is kind of happening during the telophase in there. But more on mitosis later. I think that's topic 10 or 11, 12. I can't remember, one of those. 12. Okay, so let's talk about the cell wall. We've already talked about cell walls, but just a reminder that the cell wall is made out of peptidoglycan. So here's our peptidoglycan structure again. I have a little video here in the corner. I'm going to see if it's going to play, and she's just going to kind of review peptidoglycan structure. It's a very short video. I think it's like 40 seconds long. So I'll press play on that. Sorry, I'm just going to make sure you can hear the sound. I'm going to restart that just in case.
Cell its shape and strength and counteracts in layer over the inner cell membrane. Peptidoglycan is a polymer whose molecules are cross-linked by amino acids to form a protective grid over the cell membrane. The peptidoglycan layer gives the cell its shape and strength and counteracts internal osmotic pressure from the cytoplasm. Without peptidoglycan, the cell would rupture and die. In gram-negative bacteria, the peptidoglycan layer is much thinner and is located in the periplasmic space between the inner plasma membrane and an outer membrane.
Okay, that's it for that little video. So we're going to talk a little bit about these two types of bacteria, the gram-negative and the gram-positive. That will be in next week's lab. We're going to talk about the gram stain. So you can see there's a diagram there of a gram stain, and you can see two different species on there. One species is staining the darker purple, and one species is staining the lighter pink color. So remember, a gram-positive is the one with the thick cell wall, and the gram-negative is the sandwich one that has the two membranes and the thin layer of peptidoglycan. So what is going on here with this? So I want to talk a little bit about how this works, and again, we'll talk about it in the lab next week. But basically, the gram-positive stains well with the purple. The crystal violet, it will stick to the cell. It gets stuck there in that thick peptidoglycan layer. In the gram-negative, the crystal violet, it'll stick, but very loosely, and as soon as you wash, the crystal violet will actually wash away. And so we stain with a second stain, which is called saffron and red, which is kind of a pinkish reddish color. So like I said, more on the gram stain with next week's lab. Oh, I actually do have some information here, so I'll just go over it quickly. Actually, no, you know what? I'm gonna save this for the lab for next week. So there's some pictures of some gram-positive and some gram-negative cells. So we've already talked about Bacillus anthracis. You can see it's there on the left. It's the purple color. And you can see E. coli, we've already talked about as well, is a gram-negative, and it is on the right. So it's staining pink. There's a couple other species there. We've talked about Staphylococcus aureus. So remember, staph means cluster. Coccus means sphere. And if you look at those cells, there are little clusters of spheres. They look like little grapes because they stain purple. And remember, aureus means gold, the color of the colonies on a petri dish. On the right, you can see there's one other gram-negative organism. This is Neisseria gonorrhea. And you may have guessed from the name, yes, this is the organism that causes gonorrhea, so a sexually transmitted infection. So I'm showing you this slide to show you how different these things are, and actually gram stains have been around for something like 150 years, and they're still a really important diagnostic method. A lot of cases people will have, they will have strep throat, or they will have gonorrhea, and those kind of infections, they would take a swab, it gets sent to a microbiology lab. They will often grow up the culture and do the gram stain the next day. And if you get something that looks like that and is found on your genitals, that's gonorrhea. It's a very easy diagnosis. They don't really need to do any fancy tests, and they'll prescribe someone with antibiotics. Streptococcus is purple, so it will, and it's a coccus, so they're little spheres. So sorry, that's red, but strep means chain, and so Streptococcus looks kind of like this under a microscope. You've got these little chains of spheres under the microscope. So same thing. Often that leads to a quick diagnosis and then some prescription. So as I mentioned before, just to reinforce, gram negatives have unique features in their cell walls. One of those features are those lipids that are found in the outer membrane. They're called LPS or lipopolysaccharides, and they're distinct from phospholipids. And so that's just something worth noting because our immune system does not like those things. So if you end up with bacteria in the blood, sometimes that can be very dangerous. You can get fever and low blood pressure just from your immune system recognizing some sort of lipid that is not human. Other thing that I pointed out last day, of course, was the periplasm, which is kind of that unique region between the two membranes. And the periplasm has extra enzymes in there, and some of those enzymes are involved in drug resistance. So something else that we're interested in medically for bacteria. Okay, so I'm going to make a note about peptidoglycan in a moment. I just want to mention, we've been talking about these gram negatives and these gram positives and gram negatives, gram positives. Those are just kind of most bacteria. There are a few other groups, by the way, that are not considered gram negatives and gram positives. And I just wanted to show you this as an example of one of them. This is called Mycobacterium tuberculosis. So this causes tuberculosis. The myco in Mycobacterium actually stands for this thing here, mycolic acid. So you probably don't know what a mycolic acid is. It's a very waxy chemical. So what we have here is a cell that has, you can see it's got a normal plasma membrane. It's got some peptidoglycan. It's got some other carbohydrate here called arabinoglycan. But this mycolic acid, this is very important because it's very waxy and it makes it hard for the immune system to actually grab onto these cells. So what happens when somebody gets tuberculosis is the bacterium ends up in the lung, the immune system tries to grab on, and they can't destroy the cells. And so the cells will slowly grow and eventually cause disease. So I'm not really going to say much more about tuberculosis. You can always ask me about it any time, but very fascinating organism, Mycobacterium tuberculosis. So let's go back to our notes here. Don't want to forget to add peptidoglycan. So here we go. We got peptidoglycan. And what is that made of? It's made of carbohydrates. Plus it has peptide cross linkages. So what do I mean by cross linkages? They're the little short peptides that are linking the carbohydrates together. The term is cross linkage. So what is the function of peptidoglycan? This forms the cell wall, gives structural strength, I'll say, structural strength, and shape to the cell wall. So without peptidoglycan, it's really important for bacterial cells, particularly the ones that do not have, particularly ones that want to be different shapes, they need the peptidoglycan. One other question is why they have different shapes, and that's really complicated. I don't think we actually know the answer to that. Okay, back to the PowerPoint. Oh, there's a picture of Mycobacterium tuberculosis, by the way. It doesn't gram stain, by the way. So you're not getting the purple-pink stuff going on here. Instead, you're using a different stain. You can see it has a name. It's called the acid-fast stain. So there's some acid in there. And you can see this stain is a blue fuchsia stain. So fuchsia is kind of like a really vibrant, strong pink color. And you can see this is a sputum stain. So a sputum is something that somebody has coughed up. And those blue ghosty things, those are the human cells. And the bacterium is found there at the end of the arrow. Okay, so we have a matching question here around cell walls. Okay, so let's think about our cell walls for a minute here. And I have some, you can see I have some descriptors on the right and the cell type on the left. So let's go through these one by one. So animal cells, remember that animal cells do not have a cell wall. So let's look at our answers. Chitin, nope. Peptidoglycan, okay, that would work. Animal cells never have peptidoglycan, but let's just keep reading. Two membranes, I don't think that applies to this one, but our mitochondria have two membranes. But anyway, we'll keep looking. May have an extracellular matrix. That is the one. Right, remember animal cells, some of them have an extracellular matrix, which might include things like bone or connective tissue. And that's the one we are referring to here. Okay, plants. Chitin, no, that would be fungi. So we will connect fungi to chitin. Never peptidoglycan. I'm going to get back to that one. That's not what I mean by plants. Two membranes, no, unless you're talking about the chloroplast. Animals cannot digest this. That's what we're talking about for plants. So why do I have that? Because we had talked about cellulose. And cellulose, of course, is what the plant cell wall is made of, and animals can't digest it. There are some animals that do, of course, but they're not doing it themselves. They're doing it with the help of bacterial species. Okay, so we have archaea, gram-negative, gram-positive left over. Two membranes, so that it should be an easy one by now. You should know that gram-negatives have two membranes. And this last one here, gram-positive stain purple or violet. So archaea never peptidoglycan. So what does that mean? It means that archaea don't have peptidoglycan. But remember that archaea might have pseudopeptidoglycan. So sorry about the confusing terminology. I didn't name the carbohydrate in archaea. It's not peptidoglycan, but somebody thought it kind of looks like it. So they gave it that name. So sorry about my handwriting here. I'm trying to be neat. I mean, I'm much better on a whiteboard. The screen is just not easy to write on the screen, but I'm sure you're not suffering as bad than in Soren's classes. I've seen his handwriting. It's atrocious. Okay, so let's talk about some other bacterial parts. I'm calling them appendages, you can see here. And the first ones I want to talk about are called fimbriae or pili. So pilus would be one, and pili would be many. So what are these things? You can see they're shown all over this little cell here. They look like these hairs or really skinny little fingers. And that's actually what they are. They're little sticky attachment devices. So bacteria like to stick to surfaces. Sometimes they do that when they're causing an infection. Many times they do that environmentally. They're sticking to rocks and trees and plants, and you can imagine. And so these fimbriae are basically sticky little proteins. They have two names. You can see fimbriae is one name and pili is another. And those terms are used interchangeably. So you do need to know both terms. Sometimes, you know, like if you discover it and you're studying this organism, then you get to call it something, right? For example, there's a pili that's used for attachment to your bladder. So if you've ever had a bladder infection, then the organism that infects your bladder has something called a P. pilus. I don't know if that's a coincidence why they use the letter P, but it kind of sounds like P, like urine. Easy way to remember what it is. I like the word pilus better. It's easier to say, and it helps me remind myself that these things are made out of proteins, right? So P for pilus, P for protein, and so they're used for attachment. You can also see that note at the bottom. It says there's something called a sex pili or sex pilus, and these are used for genetic exchange. So I'll show you a picture of that, and then we'll go back and we'll make some notes in a minute. Here's another picture. You can see nice, really nice electron micrograph. They've added some vibrant colors to really make these things pop out. So what do we have here? This thing on the left has all these fimbriae and pili, and then it has this extra long one. And so this extra long one has a few different names. You can see we're calling it the sex pilus or the conjugation pilus. And this is involved in a process called conjugation. So what is conjugation? Maybe you've watched a prison movie or a prison TV show and they talk about the conjugal visits. You know, that's usually where a spouse comes in and they you know, the man and the woman are having some private time together, right? So conjugation means sex, basically, right? So what are we talking about here? Bacterial sex. If you read some of the textbooks, it actually sounds really exciting. They're kind of talking about this pill as being like a grappling hook. So like I said, kind of sounds exciting, right? You've got this sticky, long grappling hook-like device. It goes out and grabs another bacterium and reels it in, and then they exchange DNA. So that's basically what we mean biologically by sex is DNA exchange. I want to try to show you how this procedure works. So you can see there's two cells there, and notice they have names, right? Geneticists like to do this. So they talk about something being positive, meaning it has a gene. So in this case here, it's the F gene. So thankfully, in this case, the geneticists have named it and given it a good name because the F means fertile. I know everyone's thinking about all these F words, but I'm trying to teach you some good F words. We've got fimbriae, we've got flagella, we've got fertile. These are all good F words. So something to bring home today, some good F words. So F means fertile. So what's going on with this fertile cell is it has this plasmid here. So this plasmid has the genes for the pilus in it. So the genes that say, hey, make a pilus. So it makes the pilus, sends it out, apparently like a grappling hook, according to one of my textbooks, and it attaches to another cell in the environment. And that other cell may not necessarily be the same species. Now notice the other cell is F negative. It doesn't have to have the gene. It can just be some other cell, some other species. So there's another micrograph showing them in the act, catching them in the act of conjugation. So number two, you can see somehow the cells come closer together. So that grappling hook analogy is pretty good because you can imagine the one cell reeling in the other cell. And then what happens is the DNA strand from one is transferred to the other. So remember, DNA is double-stranded. It's a double helix. So what it does is, I don't exactly know how this works, but somehow it strips off one strand, and one strand goes from the F plus cell to the F minus cell. And then, so now they both have the genes. So both of them are now F plus, both of them are fertile cells, right? And so if there was something else on the plasmid, for example, like antibiotic resistance, now that resistance is being spread from cell to cell and maybe even in some cases species to species. So this is something we're very concerned about is this conjugation procedure. Like I said, medically it's very important when we're concerned about antibiotic resistance. Okay, so I'm going to write a note about the fimbriae and the pili. So here we go. We got fimbriae and pili. And remember, pilus means one. A lot of these things, I is usually plural, and us is singular. So what are these things are made of? These are made of proteins. And what is the function? The function is attachment to surfaces. So for example, I gave you the example of the human bladder, so we'll just say human tissues for pathogenesis. And of course, we also talked about sex pilus, and that is used for conjugation. So conjugation meaning that whole procedure where they have the DNA exchange using the F plasmid. Okay, so back to the PowerPoint. So there's actually, this is really, really recent where somebody used some really amazing fluorescence to show this sex pilus actually extend and contract. I don't know a lot about this study, but I was able to find the video online. I thought it was pretty cool looking. Okay, so let's talk about motility. So what does motility mean? It means moving around or swimming or something like that. And you can see these bacteria here, they're kind of swimming around. And they're swimming around in kind of a weird fashion. And so what is going on here? These things have yet another F word, flagella. And flagella are like tails, except for these tails, they don't flap up and down or back and forth. These tails rotate like propellers. So you can think of it as propeller like a motorboat, right? And they'll go clockwise and counterclockwise. And what you're seeing in the video is that as it goes from clockwise to counterclockwise, they change direction. So if I go back to the video here and press play, so you can see that they're kind of zigzagging around, changing direction. And basically all that's happening is they're changing the direction of their flagella, and that's allowing them to change direction. So flagella are also made out of proteins. Oh, there's a little animation showing it rotating around and around and around. These are really fascinating. You've got a very, very small motor is what you have here, a cellular motor. And we're going to talk more about flagella in topic 7, I believe. And we're going to compare it to the eukaryotic flagella. So stay tuned for that. So notice from this diagram here, you can see that the flagella is embedded directly into the cell membrane. We've got a gram-negative here. It's going right through the peptidoglycan layer, and it's getting anchored there. So like I said, we're going to come back more on the eukaryotic flagellum in topic 7, but I'm just trying to emphasize right here that they have the same name, flagellum, but they're very, very different structures. They're used for swimming, yes. The prokaryotic flagella, they'll rotate. The eukaryotic flagella, they kind of move back and forth like big whips. So they're very, very different structures. So sometimes flagella are used to characterize these organisms, right? They're not easy to see under a microscope. They usually need special stains, but sometimes that is helping people with understanding what's going on with the biology of the organism. E. coli, for example, tends to be flagellated. So that's the word meaning it has lots of flagellas, and that's a characteristic of E. coli. I think I have some more pictures here for you. Oh, there's a cartoon. Oh, the far side. And she says, he told you that? Well, he's pulling your flagellum, Nancy. So anyway, sometimes, you know, people see these far side cartoons and they don't know what is Gary Larson talking about. Now you know. Now you might get this joke. So there's actually a really cool video there on the left. And again, using some fluorescence, and you can actually make out the flagella rotating. So I thought that was really cool. I already showed you the video on the top, but the one on the bottom I wanted to show you because these things are tethered. That means they're attached to the glass on the microscope slide. So they can't move anywhere. So their whole bodies are actually rotating back and forth rather than them moving because their flagella are actually attached to the glass. I thought that was a pretty cool video. I'll just play it again one more time. There we go. Okay, so I'm going to go back to the Word document, add our next F word, which is flagella. And these are made out of proteins as well. So what is their function? The functions are motility, which is just a fancy word for meaning swimming. Okay, so back to the PowerPoint. And let's see here. Oh, there's one other type of flagella I wanted to show you. So take a look at this organism here. Look how it's swimming around. The entire organism is corkscrewing. And so this particular organism is something called a spirochete, which means it has a spiral shape to it. And it has something unique called an endoflagella. So rather than the flagella kind of being outside of the cell, the endoflagella is stuck there basically in the periplasm, so between the inner and the outer membrane of this gram-negative organism. So the whole organism itself actually corkscrews and moves around. This organism here, Borrelia, this is the organism that causes Lyme disease, and its ability to kind of corkscrew actually helps it to invade new tissues. So you may not know a lot about Lyme disease, but people who have Lyme disease long-term, it's starting to invade all sorts of tissues, and it can be really bad if it gets into your heart or you know, other critical tissues. So 2 words, 2 new F words for you. We got Fimbriae and we have flagella. So remember those words. Okay, so I'm just looking at the clock here. We got a few minutes left. I did have a Kahoot for you, but I think I'm just going to have to save that till next day because we'll probably run out of time here. There is another term I want to give you, and this is the glycocalyx. So glyco, remember glyco means carbohydrate, right? So in this case, I'm going to translate that meaning sugar. And calyx means coat or covering. So what is a glycocalyx? A glycocalyx is a sugar coat. So on these slides here, you can see it's representing 2 types, something called a capsule and something called a slime layer. So sorry, my writing is covering the slide a little bit. I didn't mean to do that. So what is a capsule and a slime layer? So carbohydrates chemically, they tend to absorb a lot of moisture. It's true. You can take a spoonful of sugar and you can actually add quite a bit of water to that sugar, and the sugar, you know, if you have it kind of in a pasty form, you'll notice it's actually kind of slimy. So that's what these glycocalyx are. Is they're kind of they absorb a lot of moisture. They're kind of sticky, and so these terms here, capsule and slime layer, are used somewhat interchangeably. Really, just kind of, again, it's if you are studying it, you get to name it. But generally, capsules are more neatly and tightly organized, and slime layers are kind of more goopy and spread more. So organisms that have capsules and slime layers, you actually see this on a petri dish. The cultures are just, they're slimier, they're gooier, and that's because these carbohydrates are getting shed off the outside of the layers of these organisms. So here's a picture of a capsule or slime layer, and this is allowing this organism, this is the organism that causes strep throat, and you can see it's attached to somebody's tonsil. So what are capsules and slime layers doing? They are sticky. They're allowing these things to attach to all sorts of surfaces, such as your tonsils, which is not good if you get strep throat, not fun at all. And they're also, because they're slimy, it makes it a lot harder for immune cells to grab onto them. So it allows them to evade the immune response as well. Okay, so I'm going to stop there for today, other than I'm going to fill out this Word document for glycocalyx. So glycocalyx, and I'm going to put that we have capsules and slime layers. And like I said, that's kind of a good place to stop for today and not get into anything new. And these are made out of carbohydrates, or you can call them polysaccharides if you want. So big polysaccharides, and there's a whole bunch of different ones out there. Now, what is the function? Again, we have attachment to surfaces, and we have evasion of immune cells. So evasion means escape or eluding capture or something like that. You can look it up in a dictionary. You know, that being said, right, if there's any words here that you don't know what they are, dictionary.com is a good place to start. Even Wikipedia is not too bad. You know, we have Google out there. You know, nowadays it's very easy for you to figure these things out. It's really important that as students that you do learn new words. And, you know, use these words with your friends, right? You know, talk about the glycocalyx and the capsules and the slime layers and make them part of your vocabulary. And that's going to help you learn them for this class and get to know them for future biology classes. So I see we're at the end of the time today. So thanks for coming out, and I will see you on Thursday, and maybe some of you I'll talk to tomorrow regarding the lab. So just a quick reminder that lab 1-2 report is due tomorrow. So if you have any last minute questions about that, send them my way. I will see you guys later.